Hello students, after seeing plasma membrane, today we are going to talk about the cell wall. So, plant cell in addition to the plasma membrane have another rigid outer covering called the cell wall, and the cell wall lies outside the plasma membrane. It means outside the plasma membrane there is one also covering layer which is rigid in touch is present, and that is considered as the cell wall. So the plant cell wall is mainly composed of cellulose, as we all know, and cellulose is a complex substance and provide structural strength to the plant. Now when a living plant loses water through the osmosis, there is a shrinkage or contraction of the content of the cell away from the cell wall, and this phenomenon is known as plasmolysis. So basically, suppose this we are considering as a cell wall, so the content of the cell will be nearby this cell wall like this way, right? But when the cell losing water through the osmosis process, the content of the cell will goes away from the cell wall because of the shrinkage of the cell content, right? And that procedure is considered as the plasmolysis. Now, cell walls permit the cells of plant, fungi and bacteria to withstand very dilute, means hypotonic external media without bursting, right? So in that way, so what happened, due to the osmosis process, the cell will take up the water by osmosis and the cell will start swelling up, right? So this swelling up apply the pressure on the wall of the cell, right, and it will protect from bursting out the cell. So in such media the cell tends to take up water by osmosis and the cell swells. Obviously if a cell will going to take up the water through the osmosis process, so the size of the cell wall will going to be increases. It means we can say that the cell will going to be swells, building up pressure against the cell wall. The wall exert an equal pressure against the swollen cell, and because of their walls, such cells can withstand much greater changes in the surrounding medium than the animal cell, and that is the reason that cell will not going to be burst out by taking up water and applying pressure on the cell wall. It will withstand, right? So that is the another advantages of the cell wall. Now after cell wall, let's start with the nucleus. So we have in previous video we have seen the mounting of the onion peel, right? It means we have prepared the slide for the onion peel to observe the cell of the onion, right? So in that we have observed certain thing. We had put iodine solution on the peel, that is the staining agent जैसे मैंने आपको बताया था. So why? What would we see if tried observing the peel without putting the iodine solution? So try it and see what the difference is further. Further, when we put iodine solution on the peel, did each cell get evenly colored? So that is a staining agent. As I already mentioned in the previous video, that the cell is generally transparent and onion peel is also a transparent layer. So you are not able to observe the cell under the microscope without staining it, right? So after staining it, what happened? You are able to observe the cell. See, I am saying that you are able to observe the cell, but cell organ layer we are not able to observe under a simple microscope. For that we will require a electron microscope. According to their chemical composition, different regions of cells get colored differentially, right? Some regions appear darker than other region. Apart from iodine solution, we could also use safranin solution or methylene blue solution to stain the cells. So basically if you are going to use any of the staining agent, then what happened, that each and every cells will get different color, right? See, according to its chemical composition, and different regions of the cell get colored differentially. Some of colored darker, some of lighter, right, like that way. And these are the staining agent that we can use most probably. The nucleus has a double layered covering called nuclear membrane. See, just like around the cell there is a plasma membrane, or we can call cell membrane. Exactly same way around the nucleus there is a double layer covering which is called nuclear membrane. So the nuclear membrane has pores which allow the transfer of material from inside the nucleus to its outside, right? Just like a plasma membrane, it will also allow the exit and entry of the material from inside the nucleus to the outside the nucleus, right? Okay, that is to the cytoplasm. So it will take the material from the cytoplasm, or it will exit the material to the cytoplasm. So the content around the cell, except cell organelle, it is considered as the cytoplasm. The nucleus contains chromosomes which are visible as rod-shaped structure. See, inside the nucleus there is a presence of chromosome, and chromosome is look like a rod-like structure. So chromosome is just like this way. It is a rod-like structure, like this way. Okay, it is not a perfect, but you can have the idea of the structure of the chromosome. It is present in a pair, so that's why I have represented over here 2 chromosomes. The nucleus contains chromosomes which are visible as a rod-shaped structure only when the cell is about to divide. When the cell going to be divided, at that time the chromosome will look like this. Chromosomes contain information for inheritance of characters from parents to the next generation in the form of DNA, right? So chromosome carrying the genetic information, which we are generally calling it DNA, that is the deoxyribonucleic acid. The full form of DNA is deoxyribonucleic acid. Chromosomes are composed of DNA and protein. Mainly the structure of chromosome is composed of DNA and protein, right? DNA molecules contain the information necessary for constructing and organizing cells. DNA, it is a kind of genetic information which is passed from the parents to the next offspring. Functional segments of DNA are called genes, as I have told you. In a cell which is not dividing, this DNA is present as a part of chromatin material. See, in some of the cell is not going to be divided, right? In that case, the DNA will present as the part of chromatin material. So chromatin material is visible as an entangled mass of thread like structure. इस तरीके से chromatin will like this way. इस तरीके से chromatin structure आपको दिखेगा. It is a thread like structure, and right, it is a thread like structure got entangled with each other, right, like this way. Okay, so whenever the cell is about to divide, the chromatin material gets organized into chromosome, right? So this chromatin material will get organized into chromosome when the cell division will going to be take place, right? So the nucleus plays a central role in cellular reproduction, the process by which a single cell divides and form 2 new cells, right? Because during a cell division, this process will going to be takes place. A single cell will be divided into 2 new cells. It also play a crucial part along with the environment in determining the way the cell will develop and what form it will exhibit at maturity by directing the chemical activities of the cell. So that is the another function of the nucleus, right? That it will going to examine by directing the chemical activities of the cell. Now next, in some organisms like bacteria, the nuclear region of the cell may be poorly defined due to absence of a nuclear membrane. See, in some of the organisms, the nuclear membrane is absent. So in that case, the nucleus is not going to be well developed. So such an undefined nuclear region containing only nucleic acids is called a nucleoid, right? There are any organisms in which the outer covering or nuclear membrane is not there. So in that case, nucleus is not well developed. It is just like a nucleic acid. So that can be considered as the nucleoid. Such organisms whose cell lack a nuclear membrane are called prokaryotic cell. You already studied in the lower standard the types of cell, that is the prokaryotic cells and eukaryotic cells, right? So in case of prokaryotic cells, the nuclear membrane will be absent and the nucleus is not well developed, and it is considered as the nucleoid. Organisms with cells having a nuclear membrane are called eukaryotic. So opposite to prokaryotic, If any organism is having a nuclear membrane around nucleus, then it is considered as the eukaryotes. So prokaryotic cell also lack most of the other cytoplasmic organelles present in eukaryotic cells, right? So eukaryotic cells में जो भी organelles present होते हैं, it might be absent in the prokaryotic cells because of not having a well developed nucleus. Many of the functions of such organelles are also performed by poorly organized part of the cytoplasm, as the absence of such organelles will be performed, whatever the function will be performed by poorly organized parts in the cytoplasm of the cytoplasm. The chlorophyll in the photosynthetic prokaryotic bacteria is associated with the membranous vesicles, but not with the plastids as in the eukaryotic cells. So that is one more difference between prokaryotic and eukaryotic cells, that the chlorophyll in the photosynthetic prokaryotic bacteria is associated with the membranous vesicle, right, which is a bag-like structure present inside the cell, but not with the plastids, right, because in a eukaryotic cells the chlorophyll is associated with the plastids, but in prokaryotic cells it is associated with the membranous vesicle, which is a bag-like structure. So that is the difference between prokaryotic cells and eukaryotic cells.